Friday filled first and then we added Thursday. So I let people kind of go where they wanted. And Friday, I think more people found convenient, which I don't understand myself, but I don't find Friday afternoon pleasant for anything. So I do have a pretty big group there, but I also have a number of people that have figured out that they don't really need to show up for lab. They can either watch the video or just hand in the stuff. And that's helpful, but oftentimes they're not taking the quizzes. So you can only do so much. Is what it is. A lot of people also, most of this class is using it as a stepping stone in science, but most of the other class is not. So they are just concerned with the getting by. They just need to get by and have it as a class that they pass so that they can get a degree. And I understand that. So, but I think science is way too much fun for that. All right. Well, we will.
I have a question about the schedule.
Yep.
So Wednesday the 16th, we do or we don't have class?
Nope. Wednesday the 16th is the first day of finals. And so what happens is the last day of class is Monday. Tuesday is what we call a reading day. It's a day that they give you to study or whatever. And then finals actually start on Wednesday. And so depending on when your class is, that determines when your final is offered. And ours is offered Friday morning.
Okay.
So,
all right.
But nope, we have nothing on Wednesday.
Okay, thank you.
Well, that was an easy question. Next.
The 2 hour, it's a 2 hour final.
Yep.
Do you think that we would need most of that time?
Well, I usually try to make it 100 questions.
Okay.
So it depends. If when we do 50 questions, if you use up that entire time, then I would plan that that's probably the case. But if you find that you don't need it, the same thing's going to apply with 100, I would think. I haven't made it up yet, so I'm not guaranteeing it's 100, but there's so much to choose from.
Do you usually do multiple choice or some like written?
I keep the final all multiple choice. Okay, so, oh, that, I gotta send out something to my one. Thank you very much for reminding me. Remember when we did, what was it? It was section 2 when we did Punnett squares, and I have Punnett squares on my bio 131 for tomorrow. So I want to make sure they bring pencil and paper or have it available. All right, what do we have for questions? That easy, huh, guys? All right, well, have a great day.
Well, I have to admit I didn't make class yesterday, so I haven't watched the recording. So yet, so I
Don't you like it when someone has to admit that they did that? You really didn't have to admit that, Lisa, but
yeah, well, I did. But yeah, so I don't know what we covered yesterday, so I don't know if I have questions on it, but
Dante, you've got your answer. Maria took care of that. Okay, well, I'll tell you what I'll do. I will share the screen and I'll go into the PowerPoint. Okay, and that way, see what I'm doing here. And I'll just go through it as far as slide by slide, and stop me if there's something that you want me to go over or that reminds you of a question or anything. Okay. And we'll try it that way, unless somebody's got something they know right now they need. So you're good with parts of the heart and the vessels of the heart? I don't have any problem stopping at any of these, you guys. So if you do have questions, please ask.
Can you go over the last slide, the one before that, please?
Yep.
Can you go over this, please?
Do you want this part, Rose? The
Yes.
vessels and or this part over here that talks about the layers?
The vessels.
The vessels. All right. So remember, the heart has 4 chambers. And so we have the 2 atria that are at the top. And then we have the 2 ventricles that are the bottom chamber, and they're separated by a valve called the AV valve. And so on the right-hand side of the heart, and this is the right-hand side of the heart, the inferior vena cava, the biggest vein in the body, and the superior vena cava come into the right atrium, and they pass through the AV valve, the right AV valve, and they go into the right ventricle, and from there they're going to the heart, or the lungs, sorry. And so they're going to go through the semilunar valve, and this is the pulmonary trunk. And then the pulmonary trunk branches off into the pulmonary arteries. And the pulmonary arteries are going to go away. They're going to go to the lungs. And the pulmonary arteries are the only vessels in the body that carry deoxygenated blood, but they're called arteries. Okay. And so these vessels here are the pulmonary arteries, and those are going to the lungs. And then the pulmonary veins are going to come back to the lungs, and those are going to carry oxygenated blood. And they're going to come back in, and they're going to empty into, the right, or the left. Okay, because it's as if it's sitting in you. So this is going to be the left-hand side, the left atrium. They'll go through the left AV valve, and in the left ventricle, it's going to go up into, and we can just barely see it here, but this is the aortic semilunar valve, and this is the aorta, and the aorta is going to service the body. These vessels are going to go to the head, neck, and the upper body, the shoulders and the arms. And then notice it turns, and it's going to come actually down behind it, although we can't see it here, into the descending aorta. And what we also don't see here is that arteries come off the aorta. The first arteries that come off the aorta service the heart tissue itself. That is where the coronary arteries come off, and they service all this heart tissue. Good?
Yes, thank you.
You're welcome. And we, of course, had talked about the pulmonary circuit and the systemic circuit. The pulmonary circuit comes off the right side of the heart, whereas the systemic circuit comes off the left side of the heart. And we know about oxygen and CO2. And also pH is regulated through the buffers in the blood. We also talked about the SA node and how that starts the contraction of the atria. So the SA node is in the right atrium, and it's going to send the electrical impulse, the action potential, around both of the atria, these green lines. The next place they're going to activate is the AV node, and the AV node is responsible for all this yellow. And these fibers that go, they're embedded in the myocardium, and they go around the ventricles. Those are called Purkinje fibers, and they're basically responsible for just carrying that message. So the SA node is going to cause the contraction of the atria. The AV node is going to spread that action potential. That's going to cause the contraction of the ventricles. So the SA node is called the pacemaker of the heart. I think that kind of covers that. We talked about blood pressure. Remember this, arteries to arterioles, to capillaries, to venules and veins. So we know capillaries are our smallest blood vessels. Systolic pressure is the pressure that occurs with a ventricular contraction. That is a more or a stronger contraction, I guess, is what I'm trying to say, because the ventricles have to move the blood from the body or out into the body. So it's a stronger contraction. Even they also need to move it out into the lungs, whereas all the atrial contraction is just moving blood into the ventricle. Okay, but systolic pressure is the higher pressure. It's the one on top. Diastolic pressure is the lowest. That's when the heart is completely at rest for that half a second that it's at rest.
What's in red, is that like called something, or do we just need to know the order?
You just need to know the order. Yeah, I don't know if there's a name for that. That's a good question. Maybe I ought to give it a name so I can refer to it. But no, not that I'm aware of. We know these risk factors. We can pretty much put those risk factors down for about everything. We talked about hypertension. Normal BP is less than 120 over less than 80. Hypertension is considered over, and actually now it's changed. So hypertension is in 2 stages, and the first stage is going to be over 130, and then 80 to 90 on the bottom. So I would probably not ask you what hypertension, I know I didn't on this one, I wouldn't ask you what hypertension is considered. But it is a silent killer. That's one of the problems with hypertension. There is a lot of reasons someone might have hypertension, but it is serious if you do, should be treated, because there are no outward signs. So many people who have hypertension wouldn't know that they have it, but it does put you at increased risk of strokes and heart attacks, and messes up kidneys and that kind of stuff. What's up, Addie? We also talked about atherosclerosis, which is the thickening of the artery or around the artery, decreases the lumen, and it is increased with LDL, which is considered the bad cholesterol, and it is a decreased risk to have high HDL. High HDL, I can tell you from experience, is a bear to, it's very difficult. Well, my experience genetically, I'm sure this is genetically now, you should be able to increase your HDL with something like exercise. I have not been successful. So even in my very good exercise days, so that's a tough one, but high cholesterol will help you. But I mean, high cholesterol, high HDL will help you. Bad is high LDL. That's bad cholesterol. Okay, yeah, we looked at this. And we talked about platelets and forming a platelet plug. We actually talk a little bit more about platelets when we get into the blood unit, but we show here platelets and what a clot forming looks like, because we talked about a clot forming when we talk about atherosclerosis and the formation of a clot inside of a vessel. So that's why that came up here. Myocardial infarction, we know, is the obstruction of a coronary artery with either a clot or with plaque. and it decreases the lumen size, makes it more difficult to get blood to the downstream area. Complete occlusion of that leads to death of the tissue. And since it's serving the myocardium, that's not a good thing for us. Treatment for myocardial infarction. We've got clot busters that we can use. We do those for stroke as well. And those are meant to dissolve clots that are obstructing blood flow. Or there's angioplasty. Angioplasty is inserting a catheter and opening the blockage. And then, of course, obviously bypass surgery would be the last choice, obviously, of those, but that happens. I don't think we did this. Did we do this one? Oh, yeah,
No.
I think
Yeah.
we didn't. I thought we might have. Bradycardia, tachycardia, did we talk
We
about
did.
them? Okay, I
We
was
did.
gonna say, atrial fibrillation, which is an irregular heartbeat. It's not one good contraction of the atria. Not as important as ventricular fibrillation. That's the one where you die quickly because obviously the ventricles are pumping blood out to the body proper and to the lungs. So without a coordinated contraction in the ventricles, that does not work well. That's the one that you have the defibrillate or the what do they call IEDs located? Isn't it? Is it IEDs?
AED.
What is it? AED?
Yeah.
Okay.
Automated external defibrillator.
Yeah, the ones that you find now in schools and hallways of places. All right. And then we looked at the different components of blood. Let me know if you want to stop and talk about anything more or whatever. I'm game. Okay. Whoop. So plasma is the liquid portion of the blood. Most of that is going to be water, but it's also going to have proteins in it. Specifically, it's going to have albumin. That's going to be the most abundant plasma protein, and that is going to be very important to maintain osmotic pressure. And we know what osmosis is, and so that's part of osmosis. It also is a transport protein. It attaches, or other things attach to it, and it allows them to go around the body. Even though things like fatty acids are not soluble in a water base, like the plasma, they will attach to the albumin and get carried. And inorganic ions, calcium, phosphorus, well, phosphorus isn't in there, is it? I don't have it in there, but bicarb, magnesium, sodium, all of those are inorganic ions that are normally present in the plasma, but we also see sugar, right? We see glucose and other sugars. Lipids, amino acids, those are all going to be present in plasma, as well as oxygen and CO2. And you might be thinking oxygen and CO2 are carried in red blood cells, and that is very true, but a certain amount of the oxygen and CO2 is also going to be just dissolved in the plasma. And our cute little red blood cells are biconcave disc that doesn't have a nucleus, lasts for about 120 days. And when we are done with them, when their lifespan is up, they're removed by the liver and the spleen, and we recycle the components, specifically iron. So we keep that so we can use it in new hemoglobin molecules. We have about 5 × 10⁻12 red blood cells. That makes them the most numerous of our cells, of our blood cells, I should say. of the cells in the blood, they are the most numerous. There we go. And the goal is for them to transport oxygen and CO2. They carry that in association with hemoglobin. Anemia is when we have a reduction in the ability to carry oxygen because we don't have enough red blood cells. And there's different types of anemia. Hemorrhagic would be if you were in an accident, for example, and you were bleeding, or you had a gastrointestinal bleed of some sort, that would be a hemorrhagic anemia. Versus iron deficiency, which is where there's not enough iron to make more hemoglobin. And so we don't make as many cells. And hemolytic anemia is a situation where within the body you are destroying the red cells. Now, we talked at the very end Wednesday about blood cells, right? Remember, we talked about antigens and antibodies, and we talked about being type A or type B. When you are given blood, let's say I am type O, so my antibodies are anti-A and anti-B. If I was given type A blood, my body would actually attack those new blood cells coming into me that have the A antigen on them, and they would lyse them. They would break them apart, and that would cause an anemia as well. Okay, so that's what lysis of the red blood cells would be. And of course, blood doping is where you go to someplace like Colorado. Well, this is normal. This is normal. You go to Colorado and you are an athlete and you work out at a high altitude. You are going to, in order to increase your oxygen carrying capacity, you're going to make erythropoietin. And remember, that's in your kidneys, and it's going to stimulate your red blood cells or stimulate your bone marrow to make red blood cells. That's a normal process. Blood doping is actually introducing erythropoietin to your body in order to increase your red cell production. Okay, and that's not good. Doing it naturally by going to a high elevation is not usually a problem for most people, but by just giving yourself erythropoietin, that is not a good plan. Increases other problems associated with having too many red blood cells. All right. White blood cells. White blood cells are called leukocytes, and their job is to fight infection. And there's several different types. We mentioned lymphocytes in Wednesday's lecture. We talked about T and B lymphocytes, so we know now what they do and that they're part of our adaptive immunity. But then we also talked about neutrophils and monocytes, which are phagocytic cells, so they eat things. And eosinophils are something that we see in people with allergies. They have a role in allergies, which isn't all the best, but they also have a role in defense as well. The term leukocytosis is an increased amount of white blood cells, and that often happens when somebody has an infection. Oh, I think I have one more. So there's my lymphocyte right here. Here is my neutrophil. And then this little funny one down here that's got the red color, that's an eosinophil. And we know about leukemia. I don't think anybody doesn't know what leukemia is, but that is an overproduction of white blood cells. And there's so many white blood cells produced in the bone marrow that it crowds out the production of normal white blood cells and red blood cells and platelets that are also formed in the bone marrow. So frequently when we see a case of leukemia, we may see associated with it, when the person is diagnosed, we may see an anemia, or they may have some bruising because they don't have enough platelets. So they may have other symptoms when they're diagnosed. And I think this is our last component that we're going to talk about, and that is the platelets. Platelets are also called thrombocytes, and there's quite a few of those as well, 300 times 10 to the 9th, but not as many as there are red blood cells. The fragments of a bigger cell, that's what they really are, is just fragments of a larger cell. We saw the formation of a platelet plug, and then we saw the fibrin strands interweaving themselves into the plug to hold the platelets together. An embolus is something that is, like, you may have heard of it as something like a pulmonary embolus. And a pulmonary embolus is a part of a clot that breaks loose from a blood vessel, and it moves to an area where it gets to a vessel that's smaller, and it can no longer go any farther. And so frequently we see that as a stroke if it goes to, for example, arterioles or capillaries in the brain, myocardial infarction if it's in the coronary arteries, and then also the small vessels of the lungs are prone to having it. Those are the three most serious situations, but those are not the only places that an embolus could lodge. This is an example of an embolus formation. Remember that our veins, because there's such low pressure and there's such a long distance to go from your foot all the way up to the heart, that they have these valves that will prevent backflow. And so we count on our muscles and we count on those valves to continue the movement of blood, deoxygenated blood to the heart. So this is normal, but if we were prone to having an embolus, and we were sitting in a plane, for example, for a huge number of hours, this might be how it would form. And it would form, this is actually a thrombosis. This is called deep vein thrombosis. And that would cause pain oftentimes, it's better to be detected when it's a DVT, they call it a DVT, than when this embolus actually breaks off and floats someplace else. All right. And you must be sick of hearing about the urinary system. Do you want me to go over the parts and pieces, or are you, after lab and lecture, are you pretty good with those?
I'm good.
You're good? Okay. And urine formation, here we go. Just really quickly, because this is along those same lines, right? The capillaries, the afferent arteriole, A before E, afferent arteriole brings in the blood supply into these porous capillaries. The efferent arteriole takes it away, but the efferent arteriole is also going to become the peritubular capillaries. So all of these, this efferent arteriole is now going to surround all of these vessels of the nephron so that it can exchange either direction items, either from the glomerular filtrate back into the blood supply. That's called tubular reabsorption. Sorry, my nose itches. Tubular reabsorption, or if it's going from the blood supply into the distal tubule, that's called tubular secretion. And things that should not be present in the glomerular filtrate are things that are large, like cells. There should be no cells present in the glomerular filtrate, and there should be no proteins present Otherwise, anything small should end up in the glomerular filtrate along with water, and then it's going to be moved through the nephron until whatever is left after reabsorption and secretion is sent into the common bile, or the collecting duct. Okay, there we go. I'm losing my mouse here. So ADH, antidiuretic hormone. We remember what ADH does? That's going to tweak the water balance in the collecting duct. Antidiuretic hormone, remember a diuretic, some people will say, well, drinking tea is a diuretic. That means you urinate a lot. So a diuretic, some people have as a treatment if they have a tendency to accumulate fluid, whereas antidiuretic hormone is going to be the opposite. So instead of actually urinating a lot, you're going to pull more water into the body and not lose it in the urine. Things that notice that almost everything in this, over 90% of it is going to be pulled back in. It's going to be reabsorbed. So tubular reabsorption, except for these are a couple waste products. Urea, some of it will be reabsorbed because it can be used, but creatinine is a straight waste product. We wouldn't expect to see any creatinine present in urine. I mean, we'd expect to see it all present in urine. Here we go. None of it will be reabsorbed. The urinary system has lots of functions. The basic ones are to control all of this, the levels of the ions in the blood volume and blood pressure. ADH is part of that, producing renin. Renin is going to also play a role in that. Okay, so renin, and we won't go into that. I won't ask you that anyway, but the end result here is going to be increased blood pressure. And by reabsorbing more water, we're also going to increase the blood volume. Also, we want to regulate the pH, and we can do that by getting rid of hydrogen ions, for example, in the kidneys. Then we also want to get rid of waste, creatinine and urea, both we'll get rid of in the urine. Besides renin, we also secrete erythropoietin to stimulate the production of red cells. Also, the inactive form of vitamin D is made active in the kidney. I think that's it. Yeah. Cystitis is basically a bladder infection. You might have heard it as a bladder infection or a UTI, which is a urinary tract infection. And if you had a bladder infection, you'd expect that you would see white blood cells, right? They would, because it's an infection, you would expect that the urine would produce, or urine would have in it white blood cells when it's examined. And you may, depending on how sick you are, you may have a general increase in the blood of white blood cells as well. But pyelonephritis is when that infection is in the kidneys. Renal failure, there's both acute and chronic, and we know all the different functions of the kidneys. So we could, if I were to ask you something about renal failure, could lead to basically any of those functions it could impact. Kidney stones and polycystic kidney disease, I don't think we really covered much about those other than probably their definition. Oh, I guess I And then, of course, we have our respiratory system. I'm sick of talking about the respiratory system, so I'm guessing that you guys are sick of hearing it. Is there anything specific you'd like me to go over on here? Well, basically the respiratory system, let's see, regulates acid-base balance and moves oxygen and CO2, right? So we kind of covered that a number of times. And we just talked about inhalation and exhalation. And then we have health issues, pneumonia, which I think everybody knows, and fluid actually accumulates in the alveoli, and bronchitis, where you're seeing inflamed bronchial passages versus the bacterial infection in a strep throat, which hurts when you just breathe. And emphysema is when we have that loss of the elasticity in the alveoli. The end result really is that instead of having those alveoli be like little bunches of grapes, where they have a lot of surface area to exchange oxygen and CO2, they end up being more like a balloon. And so we would see that damaged alveoli is going to lead to shortness of breath. And this is something that's frequently seen in smokers, not a surprise. And it's a type of COPD, which is chronic obstructive pulmonary disease. We talked about cystic fibrosis in the past when we talked about genetics and the fact that cystic fibrosis is a disease where the mucus, although there's other things as well, but primarily the mucus is so thick that it traps bacteria instead of being able to move the bacteria up in the mucus and get it out of the lungs. So that is the major problem of cystic fibrosis. Asthma is inflammation of the bronchial tubes, which leads to that chest tightness and wheezing. And we talked about viruses and how viruses work, specifically about coronavirus. And how the coronavirus attaches to cells. In the case of coronavirus, the spike protein on the coronavirus attaches to ACE2, which makes me think, let me look real quick back here and see. Right there, if you can see on, it says angiotensin converting enzyme, that's ACE. So the ACE2 inhibitor is actually a secondary inhibitor for angiotensin converting enzyme. There's a couple different ways that it gets converted. That's why you're seeing it here in the lungs. So that's an actual receptor on the cell that actually allows the infection with coronavirus, but that's the enzyme that we're talking about and where it would actually be converted on the surface of the cell. All right. And let's see. So we talked about coronavirus. We did go, oh, you're seeing some of that, but we'll do that at the end again. We talked about the endocrine system and the different parts. We did not go over specifics. I gave you a few examples to know. So the role of the endocrine system is basically to control or regulate body functions. and it's going to travel in the blood. Remember that there's a difference between an endocrine gland and an exocrine gland, and we talked about that briefly, whereas the endocrine gland secretes its hormones directly into the blood, whereas an exocrine gland uses a duct. So a salivary gland would be an exocrine gland versus the thyroid gland, which is going to be an endocrine gland. So examples, the pituitary gland secretes antidiuretic hormone, and that's going to cause the kidneys to absorb more water. We see the testes are going to make testosterone, and that's responsible for secondary sex characteristics. Oops, I can't do that way. Let's try this. Oh, that wasn't what I meant to do here. Okay, we talked about the regulation of glucose by the pancreas. Does anybody need to go over that again? Are we good with insulin and glucagon? All right, I'll say no news is good news. And reproduction. The pituitary gland secretes follicle stimulating hormone. Follicle stimulating hormone is necessary for the maturation of the oocyte. We also, one of my favorites, the adrenal glands, because they're actually found right on top of the kidneys, and they secrete epinephrine and norepinephrine, and that's that fight or flight response. So we know that that leads to our heart rate going up. Our blood sugar will go up because we want to have a ready source of energy. You'll find that your breath, you're going to breathe more quickly. Your irises are going to dilate, so you take in more of, you have better visual acuity. So, and that is associated, for way back, the autonomic nervous system and the sympathetic nervous system. And I don't think we went through this. I think we talked about hyperthyroid, and I think we talked about hypothyroid and some symptoms of each, but I don't think we talked about this on the side and really how it worked. So you can skip that. We did talk about type 1 and type 2 diabetes. And then we get to the lymphatic and the immune systems. So these should be pretty familiar since they were the last 2 days. And we talked about the functions of the lymphatic system. And the lymphatic system is part of the immune system. We consider it part of the immune system. That's why it's here, but it's a system all itself. And it's responsible for draining excess interstitial fluid back into the blood. So there is a whole bunch of water. I say water, but it's fluid. It's really kind of similar to plasma, but there's a whole bunch of it that bathes the cells. Not all of our fluid is found in our cardiovascular system. So one of the main functions of the lymphatic system is to drain that fluid and get it back into the blood supply. And that's going to basically work on keeping the balance of water where it needs to be in the body. Also, it's going to transport lipids from the GI tract. We saw how a lacteal did that when we talked about the digestive system. And it is also going to help us in our immune response. And we'll look at the immune response. Well, this is parts of our lymphatic system, parts that are considered our lymphatic system. Seems to go on forever. The vessels that they look just like the cardiovascular system, right, as they go around the body. But they also have lymph nodes periodically, and then the lymph nodes are masses of white blood cells, and those masses allow us to check out what's going on in the body. It's a surveillance mechanism. So as the fluid is going back to the heart, or the fluid is being drained along this drainage route, there's going to be these lymph nodes that are going to have a whole bunch of white blood cells in them. And as the fluid goes through, it's going to be checked for pathogens or things that are foreign invaders that are going to encourage the immune system to have a response. Tonsils are part of the lymphatic system, the spleen, and the thymus gland. The spleen, well, yeah, that's it. And we had the innate versus the adaptive immunity. And our adaptive immunity was the last part where we talked about T and B cells and how they worked, and antibodies. But the innate immunity is non-specific. And that's going to be, first of all, our barriers that can keep things from getting in. Our second line is going to be a little more specific for things that come in, but everybody has it, and it's going to be the same for every substance that comes in. So we're going to see our white blood cells that are going to eat things. We're going to see inflammation and fever. We'll see antimicrobial substances. We're going to see cytokines, those cellular messengers. And so we have our barriers, and these were all barriers and how they worked. And then we had our cytokines, which are our cell messengers that are going to carry messages from one cell to another, or from one cell to another area of the body. We talked about phagocytosis and the types of cells that are considered phagocytic cells. Phagocytic cells, we know when we talked about the parts of the cell, that if something is brought into a phagocytic cell, then a lysosome is going to put digestive enzymes on it. It's going to combine with the vesicle that's carrying that, throw in some digestive enzymes to break it down. And part of our immune response is going to be inflammation. And I know we often think of that as meaning we have an infection or something is wrong. And it can be. We can have a serious infection, but we also are going to do that because there is damage to cells, and the damage that's occurred to those cells is going to start that immune response. That's our body responding. That's not a bacteria doing that to you. It is your body in its fight against some foreign invader. The redness is going to be caused by vasodilation, and that's going to be caused by the chemicals that the cell, when it becomes damaged, is going to release. And then, of course, with vasodilation and that increased permeability to the capillaries, now you're going to have a lot of extra fluid in the location. That extra fluid is going to press on nerve endings. So it's going to be painful, and it'll add to the swelling in the area. Our 3 R's of immune responses. We need to be able to recognize whether something is foreign or not and respond to that particular invader if it is foreign. And we need to be able to remember that reaction. So all of those are our 3 R's: recognize, respond, and remember. So we talked about antigens and the fact that antigens are a foreign molecule that will trigger an immune response. So an antigen is something that triggers an antibody. And antibodies respond to specific antigens. Antibodies, so an antibody is this Y-shaped thing, and it's got this variable region here that is very specific for one antigen. Okay, so all the different antigens would have different configurations here. So this antigen, this pretty little antigen, will react with this antibody. Antibodies are made by plasma cells, and plasma cells are B lymphocytes. There's the 5 types of immunoglobulin, GMAID, and the most common ones are IgG and IgM. And we looked at immunity, and we talked about active immunity versus passive immunity. Active immunity requires your body to do something. But in exchange for your body doing something, it is long-lived because you make memories. You make memory cells that when they see whatever that is again, that they're prepared to protect you. Whereas passive means you get short-term protection, but you don't have a memory of that. So active natural immunity is when you're going to get the disease, when you actually get the disease and get better. And now you have that long-lived memory. And then artificial is also going to provide that long-lived memory, but you don't have to get sick to do it. It's a vaccine. Versus passive, which is natural. You have natural, which would be something like nursing, or artificial, which I think I gave the example that they're doing that for coronavirus, and they're giving monoclonal antibodies. Those are not going to give anybody protection after a short period of time. And we talked about this, and this is first exposure to something. So this is how we like a vaccine to work. This would be our first exposure to some little piece of that virus, let's say the coronavirus. And so our body is going to learn what that is, that's foreign. And it is going to make antibody. But then over time, when we see that again, so we see coronavirus after we've had the vaccine, and our body reacts quickly and we don't get sick. It acts more quickly and it's got a more robust response. That's why we use vaccines, is for that immunological memory. And we talked about the adaptive immunity portion, and we talked about the presence of antibody being made by B cells, right? And those are called plasma cells when they go to produce antibody. And we were talking about the fact that the antigen has to be very specific for that particular B cell. So notice that there's different shapes here in the receptors. That's antibody that's on the surface. And this one happens to trigger this particular B cell. And that B cell will now clone itself and make a lot of different copies. Some of those clones will become memory cells. others will make antibody and secrete that antibody into the blood. So that's one part of our adaptive immunity. And then the other part is cell-mediated immunity. And cell-mediated immunity is the, first of all, cytotoxic T cell, and activating the cytotoxic T cell. This is an antigen-presenting cell, so this would be something like a macrophage or a monocyte or a neutrophil. One of those cells that phagocytizes, that eats things, it's going to present that particular antigen from whatever it ate to this T cell, this particular type of T cell. That T cell is going to clone itself again, so it's going to make a lot more of itself. Some of those will provide memory, and then the others are actually going to be able to surveil. All of these extra cells are going to be able to go out and find where this particular antigen is, and they actually then rain chemicals onto whatever they attach to that's got that antigen, and they'll rain chemicals onto it to destroy it. I wish they showed that a little bit better there. But a helper cell, a T helper cell, is similar in the fact that the antigen presenting cell is going to present it, and it's going to clone itself, and then some are going to produce memory cells, but the other ones look like they really are not doing a whole lot. But what they're actually going to do is they're going to make chemical messengers. And those chemical messengers are very powerful, and they modulate that immune response. They call other components to that area. So their helper T cells are really, really important. Basically, activation of both our B and T cells requires recognition of the antigen, and then rounds of cell division that's going to make lots and lots of cells, and that's going to be through mitosis, right? Anytime we have cell division, that's done by mitosis in our body cells. And then those specialized cells are going to either do what they're supposed to do, either produce chemical or go out and seek and destroy, in the case of a cytotoxic T cell. And they're also going to maintain the memory cells. And I showed you this and how important the cytokines are in all of these functions. So our CD4 cell is this one, and this is the one that HIV takes out, okay? And we know the result of the virus with the HIV virus taking out a bunch of our CD4 cells. So this is our cell-mediated immunity, is our CD4, and we've also got a C cytotoxic T cell. There we go. All right. And lastly, we talked about blood groups. Does anybody have questions about these? Can we skip these, or you want to go over them? My experience is people either get it or they don't. So I'm willing to go over it if somebody doesn't get it, but if you guys have got it.
I'm good.
I'm good.
Skip
Good.
it.
Awesome. You good, Zelene? All right. Then we'll skip going over that. And that was basically the end. Are we good with the universal donor and universal recipient? If you're good with the blood groups?
Yeah, I am.
Okay. And that we didn't go over yesterday, but if you want to try to do that, that will tell you whether you're good with the blood groups. And then that was it. I can't believe we're done, but if anybody wants to, I'll leave that up. And if anybody wants to try and do it just to see if they're good, we can. Any questions on all that?
So the final exam, that could be anything from the beginning of the class till now, right?
Yep. Final exam is cumulative.
Is it just like the regular exams we'll have, except we'll have 2 hours?
Yep, you'll have 2 hours. You'll probably have 100 questions, and you'll be able to use your book and your notes.
Well, I'm good. Thank you, Miss Kathy.
All right. Well, it's been wonderful having you guys. You've been a fun lab. Not that I didn't like my other students, but it's a smaller lab. I like that. So I
Oh,
enjoyed the
well then.
semester, guys. Good luck tomorrow.
Thanks.
I'll see you Monday for the,
Yep, thank you.
have a good day.
You too. Thank you.
Okay, I gotta do the quiz, but I also have a question.
Yep.
Okay, so on the review sheet, I know there's a couple of them that we're not doing because we didn't go over them in class. But
Okay.
on the lymphatic and immune system, for number 2, you have name and locate the lymphoid organs. I went onto the textbook because I didn't find it on the PowerPoint anywhere, and the only one that comes up is the spleen. Is that what you're looking for that, or is it not going to be on there?
I think I probably skipped that. I wouldn't worry too much about it because we didn't label and identify any of them. So I wouldn't worry about that one. Knowing what was on that about what the spleen does, what the thymus is, that's fine, but you don't have to know where they are.
Okay. And then what about the last 2 underneath there you have for the describe the role in the immunity played by the NK cells, the cytotoxic cells, helper and suppressor? Are they all the ones? Or
No.
do you want just the T and the B ones?
The plasma cells, you'll need to know. I'm not sure if you didn't mention that, but then you can skip NK cells.
Okay.
And you can skip suppressor cells. The other 2 are the T cells, the T cytotoxic and the T helper. Those and the B cells.
Okay. So okay, because I did the whole review sheet. I was writing them down in the notebook, so like I had all the answers, but I couldn't find those because we didn't go over them in the PowerPoints.
Yep.
Okay. What about the D antigen?
We didn't
we
do
go-
that. We skipped
Okay.
it. Yeah.
But we did do the one disease for newborns.
Hemolytic disease, we didn't do that. That's associated with D antigen, and we didn't do that.
Okay. I can cross that off too. Okay, I think I found everything else on it. So
Good.
that's what I was gonna ask you. What do you recommend to do for the final exam? Do you think like the review sheets, like all the review sheets?
That would be a really good place to start. That
Because I printed out all the old exams, but I remember you saying that you're not going to use any of the old questions.
I try not to reuse the questions, but the key is if there is here's my thought, and it's not necessarily going to get 100 for a student if they go by this, but if I ask a question on the exam, then it's fair game. And it may not be the exact same question, but whatever that principle is, it was important enough for me to ask. If I don't ask it on each individual exam, the likelihood I'm going to pick it out on the final as being that important, that concept as being that important, or that detail I'm less likely to do that. That doesn't mean there isn't every once in a while a couple that end up on there just because I'm going through and I say, oh, I like that question. But for the most part, what I ask on the regular exams is the stuff I think is important. So if the concept is on the regular exam, then it's more likely to show up on the final. So studying your notes is helpful, but the review sheets and the old exams, because of those principles, are helpful, I guess.
Okay, because I tried it my best of my ability to keep like what's going to be mainly on the exams in front of me, because like then I have them right there if I need to look up an answer. They're like
Mhm.
right there. So like most of the time with the review sheets, I have a notebook with the answers to each one, and then I had the exams printed out. But then I wonder if I need my textbook or if I need anything else, probably like the PowerPoints.
How are you finding with time? Are you doing okay with the double time? Because you'll have
Not
4 hours.
like I get done very, very close to the time being up.
Okay.
So it's like
Then,
it's like a tight squeeze to get them done. Like the one time I only had like 2 minutes left, the other time I had 18 minutes left. So it's like a tight squeeze.
so then I think, and you've done well on them though. So I would say whatever you're doing is working. So if
Okay.
you've just got the review sheet in front of you and access to whatever you've been using, I think you're doing okay. Don't you?
Okay. Yeah, I think so, but it's like the finals, all those chapters. So it's like I'm gonna
No.
have more, and I'm like trying to figure out not to get overwhelmed with all the papers. So I'm trying to figure out a way to like in the 4 hours to have them organized. So it's like if a question comes up, it's gonna be there.
Yeah, I
So
know.
I'm kind of thinking the review sheets are gonna be the thing to have,
I'm
because then at least
thinking you're
I'm like
probably
at least I
right.
can look at the review sheets, and if one of the questions says like an organ or something, I know it's from the organ system, PowerPoint or whatever. So it kind of gives me a general idea.
Yeah. I can't believe we're here, but
Is the stuff from lab going to be on it?
not as such.